So it'll basically prevent the viral code from interacting with receptors on host cells, so they can't usually enter the cells.
That's correct. So how do I explain that? I'm just going to try and explain that to some of you who may not have understood that fully well. Let's see. Let's say this is a virus protein. I'm just putting it P. It's a virus protein on the virus surface. Okay. Now, if the virus is replicating in the body, this protein can elicit several antibodies in the body, not just one, but several. Like, for example, we call these epitope. Let's say this is one epitope, second epitope, third epitope. So this protein can have, let's say, like 50 epitopes, just an example. So each of the 50 epitopes can elicit an antibody response in our body. But then what happens is this, I'm just going to draw over my writing. Let's say this is the host cell here, and this is how it binds to the host cell. Right here, this is the interaction. So this little epitope here is very critical in determining what is going to be the neutralizing antibody response. In other words, if I have antibodies to this epitope, this epitope, all these are not going to help me in much at all in preventing the virus from entering the cell, or it will not prevent the interaction between the virus and the host cell receptor. However, neutralizing antibodies, for example, this little one here on the virus protein, if you have an antibody against this, it will prevent the interaction of this little epitope with the host cell and thereby prevent the virus entering the host cell. And these antibodies that prevent the virus from entering the host cell or prevent the virus from infecting a host cell, those are called neutralizing antibodies, as the name suggests. And this term is very important term in virology, not just for SARS-CoV-2, for any virus. This is a very important term, the term called neutralizing antibodies. Okay, like this is a recap of what all of you know. The virus contains a genome that can be DNA or RNA. Then it contains a capsid protein that I'm sketching out on the image. So together, so this capsid protein is a protein, right? And the genome and the capsid together, they form what is called the nucleocapsid. On the cartoon on your right, you have the genome again. You have the capsid right here. This is the capsid protein. In addition to that, you have something called the envelope. So not all viruses are enveloped, only some viruses are enveloped. And the nucleic acid and the capsid or the protein coat together is called the nucleocapsid. And the nucleocapsid in some cases may have an outer envelope in case of the enveloped viruses. So very simple definition. All of you know that. It's just a recap. So viruses have their genome. They encode their own proteins. Not too many of them, but they do encode proteins. For example, the SARS-CoV-2 that all of us know probably is the best virus, or in other words, most of us know more about SARS-CoV-2 than any other virus. So in terms of SARS-CoV-2, it encodes almost like 28 to 30 proteins in that ballpark. It encodes that many proteins, not too many. For example, each cell in the human body can encode like 25,000 proteins or even more, 25,000 genes, and they could have variants of genes. So if you compare it to the human genome or the human cell, viruses encode much fewer proteins. There are some viruses that encode as little as only 5 proteins. Okay, so viruses encode very few proteins, but they are important for virus replication. Now, if you look at the proteins encoded by viruses or the ones that they carry in their genome, so that has the genetic information for to be translated into viral proteins. And what are these proteins that the virus encodes? Obviously, there are going to be structural proteins that help protect the genome, like the capsid and the envelope and all that. They may be important for attachment because they are the outermost layer. They help interact with the host cell. And there are going to be non-structural proteins. By non-structural, they don't necessarily contribute to the virus structure, but they could be enzymes such as polymerases, reverse transcriptases, and several RNA viruses. Almost all RNA viruses encode their own proteases necessary for important aspects, because a lot of times RNA viruses make a big protein called a polyprotein, which is cleaved down or chopped up by its own proteases into separate proteins, and then they function individually. So they encode many viruses, especially RNA viruses, encode their own proteases, which means these are enzymes that can break up larger proteins into smaller proteins at specific sites so that they are individual proteins and can do the function they're supposed to do. Now, enveloped viruses, they encode viral glycoproteins, which are part of the envelope, the outermost layer. Not all viruses are enveloped again. And there could be some spikes in the glycoproteins. I'm going to show you some cartoon, and these spikes are called peplomers. So this is just an image to show you the spikes or peplomers on the envelope. So you can see that these are the peplomers that you can see in the electron micrograph as well. Now, what about the viral envelope? It contains glycoproteins that are encoded from the genome of the virus. It carries the genetic information to encode this protein. This protein is important for virus functionality, to virus replication, everything. It's important. So that's this part. But these virus envelopes, because they bud off or pinch off from an infected host cell, they contain lipids from the host cells. So virus glycoproteins or the virus envelope contains a little bit of lipids, which is usually host derived from the host cell membranes, where it pinches off, and it also contains virus glycoproteins, which are encoded by the virus. So glycoproteins are nothing but proteins that are bound to carbohydrates. So like all of you understand, because envelope is the outermost structure, it plays an important role in attachment of the virus to the host cell. For example, SARS-CoV-2, if it doesn't have the envelope, it will not infect humans. So for all enveloped viruses, not just SARS-CoV-2, the loss of the envelope is equal to the loss of infectivity. Or in other words, if it loses the envelope, it becomes useless. It cannot infect our respiratory tract after that. Same is true for all enveloped viruses. Give me a second. Hello? That's me?
Yes, sir.
गुड मॉर्निंग रामलाल जी, मैं क्लास में हूँ, 10 मिनट में 1 घंटे में करूंगा. We sorry about it. Did anybody have a question, guys?
No, sir.
Okay, let's continue.
So I
Go
have
ahead.
one question
Sure,
regarding
go
the
ahead.
neutralizing antibodies.
Yes,
So,
sure.
so one virus could infect multiple cells, and maybe for one cell there is one epitope, and for the other one there's another epitope. So,
Okay.
for one virus, we could have multiple neutralizing antibodies.
Okay, I'll answer this question like this. Typically, okay, so if you look at there are many examples. Even if you look at SARS-CoV-2, ACE2, and there are other molecules which they think are important for entry. Let's just stick to ACE2, for example. So it uses ACE2 in the respiratory tract. It also uses ACE2 in the stomach to infect some cells in the stomach. It can also use ACE2 in the neuronal cells to infect neuronal cells. It's usually the same receptor. That means it's the same part of the virus also that interacts. Does that make sense?
Right, sir.
So although a virus can infect multiple cell types, more often than not, it uses just a single receptor in a given host. And therefore, you usually have only one set of neutralizing antibodies. But sometimes actually you will not. Okay, maybe I'll give you an example. Hold on a second. Okay. The question that whether there can be multiple neutralizing antibodies. Okay, so I'll just draw a cartoon and then try to explain it to you. Let me see if I can do it better like this. Okay, let's say this is a virus protein and this is a host cell. Give me a second. Okay, this is the host cell. H for host cell, V for virus protein. Now, this epitope could be neutralizing. This may be binding to a core receptor, which is the second receptor. So you could also have antibodies against this epitope, which are neutralizing. But the bottom line is, they could be just one or a couple of epitopes which are neutralizing. But if you take a single virus protein, it may have as many as 100 epitopes, or even much more than 100 epitopes. So only a few epitopes out of several dozens or hundreds of epitopes in a given protein could be neutralizing. It doesn't necessarily have to be just one epitope. It could be a couple of epitopes. It's not going to be like a dozen epitopes usually. It's just one or two at best. It's not going to be more than that in most cases. Does that does that
Yes,
answer your
it
question?
does.
Okay. Okay, so there we go. So So enveloped viruses like SARS-CoV-2 are much, much easier to disinfect. They are easy to disinfect with the hand sanitizer because it takes care of the lipids, and the glycoproteins are much more amenable to disinfection than a virus like this. A virus like this is much more harder to disinfect because it's difficult to damage the protein coat. It's much easier to damage this envelope, which contains some lipids. Is much more easier to damage this envelope because of the lipid content. So enveloped viruses are much, much easier to disinfect, including with hand sanitizers, and they are also susceptible to the acid pH in the gut. And so most envelope viruses, they don't survive this acid pH because the envelope is susceptible to the acid pH. And once it loses the envelope, if it passes on to the rest of your stomach, it's not, or the intestine or the GI tract, it's not going to be infective because it's lost the outermost coat, which is essential for infectivity. So typically envelope viruses are easier to handle or disinfect. Okay, going on to virus nucleic acids, I told you viruses can contain DNA or RNA as the genetic material. So these are just some numbers. You don't have to know exactly, you don't have to exactly by heart these numbers, but these numbers tell you a little bit of something. So first thing, what do they tell you? DNA viruses can be much, much longer or larger genomes. They could have much larger genomes than RNA viruses. You can see that they could have a genome of like 375 kilobases as well. Like, for example, does anybody know what is the size of E. coli genome? Approximate.
Yes, sir. 6.4 MB.
Okay, so let's just keep it like 5 MB is the size of a bacterial genome, just ballpark. What is the size of the human genome?
6.4 billion base pairs.
Okay, 6.4 billion base pairs. Billion, okay? E. coli is million, humans are billion, and these are just few lakhs actually. So if this means 300,000 bases, not much at all, much less than even a million. That is the largest virus. Smallest DNA viruses can be as little as 3 kilobases in size. So RNA viruses, RNA is much more error prone, and the largest RNA virus genome that we know of belongs to SARS-CoV-2 or coronaviruses in general. They are the largest category, so about 30 kilobases. RNA viruses can have, like poliovirus, can be as small as 7 to 9 kilobases. So they come in various sizes of genome. But then what you want to appreciate is that they are much, much, much smaller than the human genome, several orders of magnitude smaller than the human genome. In fact, much smaller than bacterial genomes as well. So they're less complicated and much, much compact. So what?
So I
Yeah,
had a question.
yeah, go ahead.
So could we say, like, since the genome size of the SARS-CoV-2 is much higher as opposed to RNA genome, so could we say that that is one of the reasons behind like the high like mutation rate of
No,
SARS-CoV?
actually, the mutation rates are actually dependent on several things. So DNA viruses mutate less, much less, because the polymerase that copies the DNA genomes has something called proofreading ability, or the ability to chop off a wrongly incorporated base, take it out, put back the right base. So they can correct errors during genome replication. That's DNA viruses. That's why they're less error prone. The second thing is I'll just keep it simple. It's much more complicated than this, but I'll just try to keep it simple. RNA viruses are more error prone because RNA polymerase, this RNA pol are the RNA polymerase that is the enzyme that is encoded by the virus to copy its genome inside the human cell. So this is called RNA polymerase, for example, in SARS-CoV-2. So this is usually error prone, and usually it cannot correct the errors it makes while copying the genome. That's how the virus replicates, by copying its genome, right? So it cannot correct errors usually. But then the RNA polymerase of SARS-CoV-2 has proofreading ability. It has evolved a protein that will even correct wrong bases. Of course, errors will still occur, but it tries to correct wrong bases so that the error that occurs is not very high. Why is that important? If the RNA polymerase does not have proofreading ability and you have such a big genome of 30 kilobases, biggest among RNA viruses, it is possible that just copying the genome, it will make errors and just die. It will make lethal genomes, which are not compatible with life. So that is a possibility. So it has proofreading ability. So that despite that proofreading ability, because it's infecting millions, billions of people, and each of us produce billions of copies of the virus, the evolution is such that it still makes errors. And when a combination of errors gives it an advantage to replicate better or transmit itself better, that becomes a new variant. But we'll talk about it later. But it's not related to the size. In general, RNA viruses mutate about 100-fold higher than, in that order, 100 to 1,000-fold higher than DNA viruses. The number of errors are 100 to 1,000-fold higher in RNA genomes. Okay, we'll talk about it a bit later, because then we have to talk about many more things. But I hope that answers your question in a short manner.
Yes, sir.
Okay, so these genomes, they could be linear. Some of the DNA viruses could have circular genomes. Some of the RNA viruses, they have something called segmented genomes like this. Like we have chromosome 1, 2, 3, 4, 5, 6. They could have segmented genomes like this. For example, a good example you may want to remember for segmented genomes or an RNA virus with segmented genomes is influenza virus. Influenza virus has 8 segments in its genome. Okay, 8 fragments of RNA constitute the influenza virus genome. So they could have linear genomes, circular genomes, or segmented genomes. This is just to sum up what kind of nucleic acids viruses could have. Okay, for the rest of the class today, we are going to look at what does the nucleocapsid look like? What does the protein coat look like? It's usually symmetrical, and that's what we are going to look at for the rest of the class in today's lecture. Now, I told you that the nucleic acid and the protein coat together is called the nucleocapsid. And this nucleocapsid usually follows various kinds of symmetry. It could be something called an icosahedral. I'll tell you what that is in a minute. It could have helical symmetry. Very rarely, only one example we know, it could have complex symmetry. So most of them fall into one of these two categories. There's one exception which falls into this category, which means neither of these. So typically, most viruses will either be like this or like this in terms of their symmetry. I'll tell you what that is in a minute. Like all of you are familiar with this, and most viruses, the nucleocapsid looks like this, something like a soccer ball. If you look at it more carefully, it actually looks something like this. This structure is called icosahedral. The structure is called an icosahedron. And This symmetry is called icosahedral symmetry. Now, can you tell me how many sides are there? How many triangles are there in the structure? Shall we count it quickly? 1, 2, 3, 4, 5, 6, correct. 7, 8, 9, 10. And you're going to have this some of these on the other side also. You can easily guess that, right? You can easily guess that some of them are going to be on the other side. How many vertices are going to be there? 1, 2, 3, 4, 5, 6, 7, 8, 9, and these 3 are going to be on the other side as well. So 9 plus 3, 12 vertices is what the structure will have, and it will have 20 triangles or 20 sides. So this structure is called a icosahedron, or this symmetry is called icosahedral symmetry, and a large number of viruses have symmetry like this. This is called icosahedral symmetry. Now, if you look at one triangle, let's say we just zoom into this triangle right here, we just zoom into it. What does it look like? It will be made up of subunits like this, called the capsomeres. Capsomeres are the subunits that make up the virus capsid protein. So this one circle doesn't mean it's just one protein. It could be made up of one or several proteins. For example, 3 proteins could contribute to one circle, and 3 of them together contributing this little unit here is called a capsomere. So this capsomere is the functional unit of the virus capsid. And this is, I'll tell you a little bit more about it. Now let's go on to the icosahedral and look at what are the capsomeres that will be and how they contribute to different types of capsomeres or different types of subunits on the virus capsid. Now, if you look at this corner or vertex, it has how many subunits does it have? 5 subunits. So all the vertices, all the 12 vertices are made up of capsomeres that have 5 subunits. Okay, so and that is called a penton. That is the vertices you can see here. So this is a vertex. This is a tip 1, 2, 3, 4, 5. So there will be 5 subunits. The shaded part 1, 2, 3, 4, and 5. So this will be the 5 subunits that make up the vertex. Now, okay, if you look at this, the subunits that are inside the triangle, on the face of the triangle, not at the vertex, but on the face of this triangle, they are going to have 6 subunits. I'll show you how. So this is a vertex, another one, another one, another one. So if you look at this blue subunit inside, it has 1, 2, 3, 4, 5, 6 subunits. I'm just going to make a line so that you can see it easier. 3, 4, 5, 6 subunits. These are the capsomeres that contribute to the face of the triangle, not the edge of the triangle, but the face of the triangle. Okay, so they are made up of 6 subunits and they are called hexons. So you could have pentons that make up the vertices, or you could have hexons that make up the sides of the triangles or the icosahedral. Now, to understand the structure better, I'm not an expert in virus structure, but some very basic things. If you, for example, let me see if I can clear it. Give me a second. Okay. Okay, if you were to pass a pencil through this hole here, how many folds of symmetry, rotational symmetry would there be? So how many times would it be identical and you can rotate it?
5 times.
That's correct. So you see, 5-fold axis of symmetry. So this is 1, 2, 3, 4, and 5. So forget this line. I'm just trying to erase this line. Now, so this gives us 5-fold axis of symmetry. You rotate it by 360 by 5, whatever that number is, that many degrees. It will look the same. If you pass a pencil through that, it will look identical. Now, I'm just going to erase that again. Give me a second. If you were to pass a pencil through this hole here, excuse me, this hole here, how many folds of symmetry do you think you can have?
6-fold.
No, it'll be this and
Okay.
this. So I'm just going to cut this off. This is something that is unavoidable by using the mouse. Sometimes it's unavoidable. So you see these 3 lines. I'm just going to draw the line again. 1, 2, and 3. So if you rotate it, so why would that be same? I'm just going to draw this triangle. You'll understand it better now. So from this corner of the triangle to this corner of the triangle, and this corner of the triangle to this corner of the triangle, it would be symmetrical, and that would be how many folds of symmetry again, guys?
3.
3 folds symmetry. Now, is there any other axis of symmetry that you can think of? Rotational symmetry. So there is this triangle here, and there is this triangle here. If I draw a line here, oh, I'm so sorry, it's a bad line. So I'm just going to erase it and try to do it again. Give me a second. Something with my mouse. Okay. So if I draw a line across like this, along where the 2 sides meet, right here. Forgive my drawing is something to do with the mouse. It's becoming scratchy. Now, this triangle here and this triangle here, they meet in this side. And if you draw a line like that on both sides of the triangle, it looks symmetrical, and that will be 2-fold axis of symmetry. So that's what I have here. Through the vertex, you'll have 5-fold axis of symmetry. Through the middle of the side of a triangle, the three corners of the triangle, you can rotate it. How many degrees would this be? 120 degrees each. So that will be 3-fold axis of symmetry. And this will be where the 2 triangles meet, this one and this one. And this side is identical to this side. So 2-fold axis of symmetry. So these are called rotational axis of symmetry. So now these icosahedrals, even before these structures were discovered in virus genomes, famous architects, particularly one called Buckminster, a US architect, he designed some of these. Anybody knows what these are? Anybody has knows either one of these 2 structures? So one is called a biosphere. This is in Montreal, and this is in Disney World. This is all designed by this architect called Buckminster, and they look like icosahedral. This structure, for example, also, and he also made a house for himself that looks like this. It's basically based on icosahedral structures, although it's slightly modified for the doors and other things here, but it's basically derived from an icosahedral structure. So these structures are very efficient in terms of designing and space and strength and a lot of things. They appear to be efficient, and engineers learned it even before we understood the structure of viruses. Now, that's the icosahedral. Yeah, go ahead.
how viruses are able to make icosahedral structures. Like they have the genome gene, and there will be genes, and that will make proteins. So why is it that like they are making a specific structures like icosahedral or helical? Why don't they make some random structure?
Yeah. So this is a lot of times this is based on self-assembly. So what happens is individual proteins are made and they are able to bind to each other in a certain manner and then self-aggregate in a certain kind of structure depending on the proteins that constitute the capsomeres. They are able to assemble in different kinds of icosahedral. There are many different kinds of icosahedral also. Depending on how many subunits come into each triangle, there are many different types of icosahedral structure also. That is self-assembly. It's like it's almost like asking give me a second, sorry. Hello? हाँ जी हाँ जी
Yes, sir.
अभी भी क्लास 11 बजे खत्म हाँ 11 बजे खत्म होगा हाँ ठीक है थैंक्स ठीक okay so it's like from the zygote, why do we all come out looking like with two legs, two hands, something like that? It's almost like self-assembly there, and it depends on the configuration of the proteins that constitute the icosahedral or the capsid. Other than that, it's just self-assembly, and they make structures like that. But there are some proteins that will not make structures like that, and that will make structures like this, and this is called excuse me, helical symmetry. Now, what is helical symmetry? It's very simple. It's just nucleic acids are being synthesized and virus proteins constantly bind to nucleic acids as and when they are synthesized. In other words, if this nucleic acid ends here, there'll be some more subunits of this kind that will cover this up like this. There'll be more subunits here, okay, like this, and that will determine the size of the helical capsid. The size of the icosahedral capsid is not necessarily determined by the genome. I'll show it to you. Give me a second. So this genome size may not necessarily determine how big this is. That is determined by the proteins that constitute the icosahedral. On the contrary, for a helical symmetry, length of the nucleic acids, wherever the nucleic acids end, that is where the capsid also ends. The size of the capsid is determined by the length of the nucleic acid. In helical symmetry, that's not true for icosahedral symmetry. Now, this is the same thing. What is the last kind of symmetry? The last one is called complex symmetry. And what is complex symmetry? Something that does not follow helical or icosahedral structures. There is only one human virus that we know of, the pox virus, that has a bullet-shaped structure and it has complex symmetry. Maybe this cartoon will tell us this is the envelope, this is the capsid protein. It looks like this, and there are other bodies on the sides of the capsid protein. It does not follow helical symmetry. It does not follow icosahedral symmetry. So this is the only virus that we know of that has complex symmetry. This is just for completion's sake. There is a third type of symmetry that does not fall under one of the other two. So if you take any virus, it could be classified into an icosahedral virus without an envelope, or an icosahedral virus with an envelope, or a helical virus without an envelope. So these structures are the proteins. What you see here, what I'm drawing on red. So that is the helical symmetry. That will look like this. So this is the helical capsid. So helical viruses with the helical symmetry and without an envelope, helical symmetry with an envelope, or complex symmetry. So basically icosahedral with or without envelope, helical with or without envelope, and complex makes it to the 5 basic types of virus structures that we know of based on capsid symmetry. Now, you don't have to by heart any of this. You will know these as you study important human viruses one by one. You will at least understand some of these. You don't have to by heart this, but this is just an example of what I have there. Any questions you guys have in terms of today's lecture?
Sir, I have a
Go
question
ahead.
actually.
Yeah, go ahead.
Sir, as you said that like as everyone says that mutation is random, but what I believe is that if like in case of coronavirus, if it is mutating only in the region that is specific for like binding to its host. So it is mutating like in only in its beneficiary, right? Right. So if
No,
it is random, then it
that
could
is
have
not true.
occurred at any other region.
That is
Yes,
not
sir.
true. For those of you who are not familiar, let me make it clear. Coronavirus is only 30,000 bases long, and almost every single base has been mutated. So that doesn't mean only 30,000 mutations have been documented. In fact, each base has been mutated. Like, for example, if it's a at the 100th position, A has become C, A has become T, A has become G. So, in fact, almost 75,000 mutations have been documented in a 30 kilobase virus, 30,000 base virus. 75,000 mutations, which means each position has mutated into at least 2 to 3 different nucleotides. The possibility is one base, A can become G, C, or T. So maximum possibility is only 90,000 mutation. It has already crossed 75,000, 80,000 mutations have been documented in coronaviruses. So that is not what happens. I'll tell you why you are forced to believe like that. As a reader or a common man, you're forced to believe that only spike protein mutates faster. That's not true. Pretty much all the proteins or the entire genome mutates more or less at the same level, with some subtle differences possible because of more complicated reasons. But why do we see more mutations in the spike protein? Because they give advantages to the virus. They become what is called fixed. See, I told you 80,000 mutations are documented. Many of them are not good for the virus. They are lost in evolution. But the ones that are good for the virus, many of them occur in the spike region because they provide technical advantages to the virus. They provide critical advantages to the virus to either replicate, transmit, or to infect a new cell type. Whatever it is, they provide advantages. They allow the virus to easily latch on. So those mutations which provide advantages to the virus, they become fixed, or those mutations are called substitutions, or they become fixed over a period of time. They will be seen in a lot more viruses. So that happens in spike because that part is important for attaching to the host, and it can confer many advantages. It may confer escape from neutralization antibody, which now Omicron as Omicron does that. So it's not that only spike mutates faster. Everything mutates more or less equally. But the spike mutations we recognize, or we are able to see it better because they become fixed more often than not. You see what I'm saying?
Yes, sir. I got it.
Sure.
Thank you, sir.
So I had another question.
Go ahead. Go right
So,
ahead.
so when like people describe these mutations, so what do you consider as like the fixed template for the virus? Because if you sequence viruses from 10 different individuals, you'll get like probably 10 different sequences. So which one do you consider to be the first one, or like where you define the mutations from?
Which one you'll consider the first one? Can you say that again? I didn't get that question very
So
clearly.
basically, how do like people actually define the mutations from? Because different strains would have different sequences. So how do you like identify that originating strain where you define the like, okay, like say the 100th base pair has been mutated, but it's possible that it hasn't been mutated with respect to the original virus, but with respect to some other strain of the virus.
No, so what happens is, let's say, what we all now use as reference strain for SARS-CoV-2 is something that was isolated from Wuhan and back in December 2019, that is now serving as the reference strain. Now, any deviation from that is now considered a mutation. Now, first came alpha, which is a set of mutations, maybe about 15 mutations that conferred some advantages. So the virus could replicate faster, spread faster, and it replaced the wild type, which is Wuhan isolate. It replaced that. Then came beta, then came gamma, then came delta, which swept everything off the carpet, and delta replaced everything. Almost everything was replaced by delta because it had more mutations. It had like 20, 25 mutations, and they conferred advantages. Now, after that came Omicron, which had like 50 mutations. Obviously, it confers certain kind of advantages, especially when when the population that it encounters is a vaccinated population. Omicron confers some advantages because pre-existing neutralizing antibodies are not able to neutralize it well. So that is the advantage that Omicron has. So that is the next set of mutations. So I don't know specifically what is your question. I'm sure I can answer it if you rephrase your question.
I wanted to know like what was the exact reference strain which like the
That is
mutations
the
are
Wuhan
defined with
isolate
respect to.
that was isolated in December and sequenced in January. December of 2019 was isolated. It was sequenced in January of 2020, and that is the RefSeq, and it's the
Fine,
universal
sir.
RefSeq.
Fine, sir.
Yeah, sure. Okay,
Sir, I have a question.
go ahead.
Sir, you talked about like in coronavirus we have 70. We have found 70,000 mutations. So which kinds of mutation are you talking about? Are you talking about a single point mutation, or like if there is two mutations uh, in the DNA, then it would be like more than 70,000. In that case, if we consider.
I don't know what you mean by more than two mutations, more than 70,000. What does that mean?
So I think he meant a permutation of different mutations. So you can have like
That's what
several like millions of permutations, right?
Yeah, I almost told you the maximum possible permutation is roughly 90,000. Because at one base, it can go to three other positions. So three mutations at one position, 30,000 times 3, 90,000 maximum possible mutations. Does that make sense?
Sir, I guess like this is not exact calculation. There must be more. Like it can be millions and billions and trillions, I guess.
It can't be it can't be trillions. Maybe theoretically it can be. See, so these are 90,000 substitutions. So this is theoretically this is the maximum limit for substitutions. But the number may go up if you include insertions and deletion. And there is no limit for insertions and deletions. If you include that, the numbers can go up. But however, thankfully, this virus, for example, so far there are only some 20 or so well-documented deletions and even lesser or fewer insertions that are well documented. After evolving in billions of hosts, and in each host billions of viruses made, after that it has only 20 deletions that are well documented and less than 10 insertions which are well documented. So that means insertions and deletions are not common in this virus. So that makes that number more or less correct.
Yes, sir, maybe. Sir, I have another question. Like you said that SARS-CoV-2 is a RNA virus and it is 30 kb long. So does it remain? Its shape is like linear. Is it a linear virus and single-stranded? Its genome is like linear and single-stranded, or does it form like double strands? And like
Okay.
how does it remain?
Okay, I'm just going to use the board very quickly to answer your question. I'll use a pen. So it is something like this. It's linear and single-stranded. But when it has to copy itself, it'll copy it like this. I'm just going to put a wavy line for the genome that it copies. This is complementary to the genome. It will copy it like this. Now this will serve as template to make many of the genome copies. So it is single-stranded RNA linear, but it copies itself to the complementary strand that is also linear, and that complementary strand will now serve as template for the virus to copy its actual genomic strand many times. So this is the complementary strand that will serve as template for to copy the genome many times as it wants. Does that answer your question?
Yes, sir. Yes, sir. Sir, does it form like hairpin structures, or when it is outside the cell, like it is not inside the cell of host? So in that case, like how is it its genome?
So see, when we say linear or circular is what we define genomes as. All RNA viruses, they have only a linear genome. The human viruses, at least, they have only linear genomes. And when we say linear, they don't necessarily mean like a straight line. They will have small kinks here. They can have a kink here, which means they can form small secondary structures. All RNAs form small secondary structures. That doesn't mean they are perfectly like a straight line. They are more or less like a straight line with some kinks in between.
Yes, sir. So, like, I have another hypothetical question, but like in nature, do we found any virus which is RNA virus and double helix RNA virus something?
No, we have double-stranded RNA viruses that can infect humans also. We have viruses like that, but it's not called it's slightly different from a DNA double helix. It doesn't form a structure like a DNA double helix.
Okay, sir. Okay. Thank you, sir.
Okay, guys. If there are no more
So,
questions,
one
yeah,
last
go ahead.
question.
Go ahead.
Fine.
Sure.
So complex viruses, complex symmetry viruses do not have an envelope.
There is only one, and it does have an envelope. That's the pox virus. Let me see if it's there in my cartoon. I don't remember. Yeah, here we go. It does have an envelope.
Got it, sir.
Okay. Any other questions? Okay, guys, we'll close then. I'll send you the slides by.
Yes, sir.
Yeah.
Please
Go
send the
ahead.
slide.
Go ahead.
Sir,
What is
just
it?
I was saying that please send the slide.
Sure. I'll do that. Okay. Thank you, guys.
Thank you, sir. Thanks a lot.
Thank you,
Thank
sir.
you, sir.
Thank you.
Thank you, sir.
Thank you, sir.